So today we're going to talk about gram-positive bacteria. And so who do I mean when I say that? Gram-positive has a variety of meanings, and you really need to be clear when you use the term gram-positive. What do you mean by that? By the way, for technical reasons, gram really ought to be capitalized. They don't let you do that. That's a guy's name, so it should be capitalized. But editors don't like that. It's no longer considered reference to his name anymore for some reason. From a phylogenetic standpoint, the gram-positive bacteria are these two phylogenetic groups, which may or may not be specifically related. This branch is not very reliable. So we're talking about a couple of phylogenetic groups. Firmicutes, these are sometimes called the low GC gram-positive bacteria. These are the ones you normally think of in terms of gram-positives. Bacillus, lactic acid bacteria, staph strep, Clostridium, those sort of things. It includes some organisms that are not gram-positive in the usual sense. It includes the so-called mollicutes, that is Mycoplasmas. When I was a student, it wasn't even sure whether Mycoplasmas were bacteria at all. It also includes a variety of organisms that have gram-negative envelopes. We'll talk about those in a minute. The second group are the Actinobacteria. These organisms include the acid-fast bacteria, Mycobacterium, etc., some really common soil creatures like Frankia and Arthrobacter. It includes Streptomyces. These are filamentous bacteria. They look a lot like molds and are common in forest environments where you peel off the bark of an old dead tree and you see this white stuff. That's usually Streptomyces. Streptomyces is what gives soil that earthy, good dirt smell. And so gram-positive is those two phylogenetic groups. I already alluded to the other meaning of gram-positive, right? What kind of an envelope does it have? In general, when you think of gram-positive, you think of an organism that has only a cytoplasmic membrane and not an outer membrane. All the other bacteria that we've talked about so far, not the planktomycetes, by the way, have a two-layer envelope. They have a cellular membrane and an outer membrane. Most of the Firmicutes do not have an outer membrane. Now, one of the outcomes of this is that because they don't have an outer membrane, they don't have a periplasmic space. And so that periplasmic space is useful. There are things that you want to do in a semi-controlled environment. And these organisms use their cell wall for this. And so the cell wall is very thick, most often, and fairly open. It's like a network or like a really, really fluffy Swiss cheese. It's full of cavities and vacancies and everything. And the void space in that functions as a periplasm. Actinobacteria, some of them also lack an outer membrane, but most of them have an outer membrane. Now, we don't call it the outer membrane. We call it the mycolic acid layer. And again, when I was a student, we thought of this as literally a wax that was formed a coating on the surface of the cells, a lot like plant cells have a cuticle made out of wax. This is false. A mycolic acid membrane is a lipid bilayer, and in many ways is no different than the outer membrane of other bacteria. It's just that it's not made of lipopolysaccharide. It's made of mycolic acid instead. And so that's the second meaning of gram-positive. What kind of envelope does it have? The third kind is, if you put crystal violet on it and let it soak for a while and then rinse it out with alcohol, does it stick or not? This has nothing to do with any of the things we've talked about so far. This has to do with how thick the cell wall is. And so you're measuring thickness in cell wall. And so this is probably the least reliable meaning of the term gram-positive. It's quite common for gram-negative organisms to stain gram-positive if the cells are really old or not very happy and they have a thick cell wall. Likewise, it's possible for many organisms that are in this group to stain gram-negative because they have thin outer or thin cell walls. Some of them don't have any cell wall at all. Mycoplasmas don't have any peptidoglycan. And so the staining isn't reliable either. But in general, most of the organisms you think of as gram-positive are in these two groups. So let's start with the Firmicutes. Bacillus, and that includes all the genera that have been split away from the original genus Bacillus because it got too cumbersome. There were hundreds of species of Bacillus. And now we have things called Geobacillus and Heliobacillus and Pentabacillus, et cetera, et cetera, et cetera. Listeria is in this group, pretty important pathogen. Pasteurella, another pathogen. The lactic acid bacteria, staph. Here are the lactics. Enterococcus, leuconostoc, lactobacillus, streptococcus. Clostridium is in this group. Clostridium is an interesting genus. Clostridium, the genus Clostridium is a lot like the genus Pseudomonas used to be, where if you're a rod-shaped endospore former and an obligate anaerobe, you were called Clostridium. It turns out that that's the primitive phenotype in this group, and so there are lots of phylogenetic groups that have that phenotype, and they've had to be all split apart. Eubacterium is a lot like Clostridium, but it doesn't form endospores. The Heliobacteria are the organisms we talked about before that have photosystems. They're phototrophs. Peptostreptococcus, Peptococcus, these are organisms that live on your skin. Who else is in here? Veillonella. We'll talk about, whoops. These organisms have an outer membrane. Some of them are endospore formers, but they have an outer membrane. And Veillonella is one of the important members of the community of your teeth. Here's Syphilothrix is another pathogen. The Mollicutes then are Mycoplasmas of various flavors. These organisms are almost without exception heterotrophs. The only exceptions to that are the Heliobacteria, which are phototrophs. Endospore formation is common in this group, but it is by no means universal. And so, staph and strep aren't spore formers, right? Lactic acid bacteria aren't spore formers. But endospore formers are in this group, and in fact, all bacterial endospore formers, all real endospores, are in this group. The endospore formation occurs nowhere else in the bacterial world. They come in all shapes and sizes. The Firmicutes in general are common in terrestrial soil and sediment environments. They are common symbionts also of plants and animals. So let's talk about some of them. How about the aerobic endospore formers? Here's Bacillus. Bacillus cereus. You guys have all seen Bacillus cereus at one point or another. Bacillus cereus is a really interesting organism. Quite common. Its normal habitat seems to be the gut of insects, but it shows up all over the place. Bacillus cereus is probably the single most frequent cause of what you call food poisoning, and generally from refrigerated meat. They had stuff that grows on it, and it really hurts your stomach. I've had it before. It's awful. Interestingly, Bacillus cereus, you think of it as individual rod-shaped cells, maybe pairs, with the endospores, and that's what it looks like as colonies on a plate. But that is not what it looks like in its natural habitat, the gut of the insect. This is what it looks like in the insect. It is filamentous. And one end of the filament is attached to the gut epithelium. And these organisms grow there. Now, wait a minute, you might say to yourself, insects, guts, obligate aerobes? It turns out that most insects' intestinal tracts are aerobic. Remember that in insects, the abdomen is where they breathe. They have trachea that run throughout the abdomen and surround the gut. And so most insects have aerobic digestion in their lower gut. And that includes the insects that these things commonly live in. And actually, for a long time, it wasn't realized that these filamentous spore formers were Bacillus cereus because they just don't look like it. It was called Arthromidas beforehand. But it is, in fact, Bacillus cereus. Nice oval terminal endospores. Most anaerobic endospore formers are in the family Clostridium. Clostridia are usually rod-shaped organisms, and it's easy to tell with some amount of confidence a bacillus from a Clostridium when you see them under the microscope, because Clostridium, the endospores are bigger in diameter relative to the mother cell and cause the mother cell to swell out. So you end up with these club-shaped structures. Whereas in Bacillus, the endospore former is narrower than the mother cell, and the mother cell's shape isn't distorted. Clostridium are anaerobic heterotrophs. They are sometimes obligate anaerobic, sometimes they're aerotolerant. Interestingly, they lack the electron transport chain. That's why they're anaerobic. They grow entirely by substrate level phosphorylation, fermentation. Now, if you're a heterotroph, you have to import stuff from the environment, right? Sugars or whatever it is you're eating. And in order to do that, you have to have a proton gradient. In order to have a proton gradient, how do you make a proton gradient with no electron transport? I've mentioned this a couple of times already. You run ATPase backwards, and you pump protons out with an ATPase. That costs energy, of course, but it provides you with a proton motive force that you can draw chemicals in with and grow by fermentation. These things are important pathogens in humans, primarily because they get in an anaerobic wound or something like that and start to grow, invading the immune system in one way or another, and basically you're starting to decompose while you're still alive. These organisms, however, are also very abundant in your gut and are important parts of your digestion, particularly those who eat protein-rich diets, meat and other sources of protein. Clostridium is usually the predominant organism in your gut. Those of you who are more sugar or starch eaters will skew that more toward organisms like Bacteroides, which we'll talk about next time. Both kinds of endospore formers can produce some pretty interesting toxins. Of course, botulism toxin comes from this organism. Botox, probably, by some accounts, the most toxic substance known to man. Why you would inject that into your face, I have no idea. Interestingly, relatives of this thing, Bacillus cereus, also form toxins. And so a close relative of this is Bacillus thuringiensis, which is basically anthrax for insects. And so this organism, when it sporulates, produces an amorphous crystal at the other end of this insect toxin. And that's why you take this stuff, they call it BT, right? And you sprinkle it on your tomatoes, and the insects die when they eat it. And it's perfectly harmless to people. Again, the mammalian version of that is Bacillus anthracis, and those organisms are very closely related. Lactic acid bacteria. These organisms also grow by fermentation and lack electron transport. This thing is Leuconostoc mesentoroides. Notice in this slide that the cells only get so close to each other. They don't bump into each other because they have a capsule. You can't see it under a microscope because it's transparent. But these organisms come together and then the capsules bump and they don't get any closer. There's some light scattering. I'm not sure if that's from the capsule or if that's just from the cells in the kind of microscopy they're doing. Notice also that they're all on the same focal plane because these things are stuck to the glass with this capsule. Leuconostoc is pretty harmless. It is an important spoilage organism in vegetables primarily, but it's also involved in normal vegetable fermentations that people do on purpose. Other lactic acid bacteria, of course, are things like Streptococcus, which is an important skin symbiont and also pathogen. Streptococcus mitis is the cause of dental caries, tooth decay. What are some other important ones in that group? Lactic acid bacteria, of course, are important symbionts in people. People like to think that you can eat them, and these are common probiotics. There's not a lot of evidence that those lactic acid bacteria will take up residence in your gut, but some people use it all the time. Well, here's a Mycoplasma, also known as a Mycoplasma. This particular one, I think, Mycoplasma mobile, but it's the same thing as Mycoplasma fermentans, Mycoplasma genitalium, etc. Mycoplasma, of course, is an important STD in humans. Oftentimes, these are described as amorphous organisms. This is false. It's just that they lack a cell wall, and when you fry and dye them on a slide, they don't retain their shape. But in reality, they have this pear shape, and this end of the cell adheres very tightly to the surface of epithelial cells. That's the attachment. These organisms are quite small. They're obligate parasites, and they have highly reduced genomes. Some of the smallest genomes of non-endoparasites are in this group. Mycoplasma genitalium has only about 540 genes in the whole genome. They have an interesting form of motility. So far as I'm aware, unique to this group. You see these fibers that stick out along the neck. Those are like little legs, and they will attach and pull down and pull the organism along the surface of the cells that they grow on. Again, these things are all obligate parasites. Some of them grow on animals, some grow on plants, and some of them even grow on unicellular eukaryotes. Yeah?
So are they analogous to a pill?
So, probably. They're unique structures. They're not made of pillin, but they are certainly analogous to that sort of a structure. They don't change in length, so they're not like we'll talk about motility next week. They're not like the pili that are involved in twitching motility that extend out and then retract. But they have some kind of a motor system that they attach forward and then bend down this way. Yeah, Mycoplasma hominis is in this group. Mycoplasma hominis is a particularly important pathogen because it's really hard to get rid of. It affects primarily females. And the reason it's hard to get rid of is that it also lives inside cells of Trichomonas vaginalis. And there is sequestered away and resistant to treatment. And then you go off the treatment and the mycoplasma comes back. And the Heliobacteria in this group, I don't have a lot to say about those. They have a standard photosystem unless I am mistaken, it is homologous. It's a photosystem 1. It has a unique chlorophyll G, and lives in soil, often associated with plant roots for reasons that are unclear. Typically rod shapes. They usually form endospores, but in culture it's really, really hard to get them to form endospores. You have to treat the cultures just right to get them to sporulate, which seems kind of odd, right? You'd think that an organism would sporulate at the drop of a hat if it's in trouble. But these organisms don't do that. And so there must be something about their life cycle that they're not seeing properly in the environment. They're very efficient nitrogen fixers. They can be autotrophic. They form this usually lime, or I guess I'd call it an army green color colony. Army green to brown on plates. And so if you see that color, that kind of dark olive green in your colonies or in your non-sulfur tubes, let me know. These organisms are anaerobes and that will grow in that tube. Also in this group are the Veillonella. There's a whole group of organisms. These organisms are phylogenetically part of the Firmicutes, but they have outer membranes. So they are structurally gram-negative. Some of them produce endospores. Sporomusa is in this group, this banana-shaped cell that forms big endospores in them. And so now you've got an endospore surrounded by a gram-negative type envelope. These organisms, the Veillonellas, are best known in 2 environments. In your mouth and in fermentations, vegetable fermentations gone wrong. And so Mount Olive down the road here keeps an eye on these organisms because they're very important in you know, if you've got 100,000 liter cabbage fermentation, if it goes wrong, that's real money. So Sporomusa is a problem in that group. Pectinatus. Megasphaera is an organism that is involved in the spoilage of my favorite vegetable fermentation, which is beer production. These organisms, many of Pectinatus, Selenomonas, etc., are pretty obvious when you see them under a microscope. They're rod-shaped, but they have flagella in a row down one side of the rod. And so when those flagella are turning, it causes the whole cell to tumble end to end and move forward like a propeller on a boat. And so what you see is this X-shaped thing because it rolls as it goes, going through the water. Probably the worst hydrodynamic way to push something through the water you can imagine, but it works for them. Veillonella is a coccus. It's one of the first things that grow on your teeth when they first erupt when you're a child. They're part of the early stages of ecological succession on those surfaces. Think of baby as an ecosystem. They are most often associated with other organisms. I don't have a picture of it. But you see long rod-shaped bacteria. These things are Sphingobacteria of various kinds. And these Veillonella cocci grow right along the surface of the cell in symbiosis.